Hello everyone. Thanks for coming. Today I would like to talk about the invasive grass that is problematic in the wetland of the Great Lakes regions. It's Phragmites australis or common reed. We have been analyzing the genome of this common reed, and I would like to present what we can learn from the genome about its invasiveness. So reed is an ecologically important grass species that often defines the landscape of wetland. In its native ranges, reeds can play beneficial roles. For example, protecting the coastline from the erosion. Uh, but in the Great Lake regions of North America, reed subspecies that grows very tall and much more robust than its native subspecies counterpart. So this is the native subspecies. You can see the height is more or less similar to a person's height. This is our colleague, Keith Clay. But the invasive one, you can see that it can grow like much taller, about 2 to 3 times taller than a person. And like when we compare the seed head and the leaf and the rhizome tissues, the invasive one is always very robust than the native. And often this is blocking the passages of wildlife in the wetland and causing a lot of problems. So we can trace the origin of this invasive subspecies using the chloroplast sequence, and this invasive subspecies was originated from the Europe. So we can see that these green dots were moved to the Great Lake regions here. So these rapid invasions disturbs local ecosystems, and it costs multi-million dollars per year to control this invasion. And it can endanger local wildlife habitats and biodiversities. And one thing is that often this invasive and native subspecies coexist. So this provides an excellent system for comparative analysis of invasiveness. So our ultimate goal, one of the major reasons why we sequence the genome of the this lead subspecies is to help develop development of biocontrol measures. So far, control of the lead invasions relies on mostly non-selective measures such as like just the cutting or burning and so on. So if we could develop a system to deliver, for example, the inhibitory RNA molecules targeting genes specific to the invasive subspecies, we could control their growth in more targeted way. So that is one of the big goals. So we started to sequence the genome of the common reed, the invasive subspecies, as the representative. There are quite a lot of genomes already available among grasses, but most of them are agricultural crops like rice, like corn, sorghum, setaria. Setaria is here. And so this draft genome of Phragmites australis is the first genome representing the Arundinoideae family, which is a grass subfamily poorly studied, despite it has a very important ecological roles. So the first thing we found that the Phragmites genome contains more genes duplicated than other grass genomes. These are all other grass genomes, and we can see that about 35% of the entire genes are duplicated in the Phragmites genome. So if we compare the Setaria is a foxtail millet, a portion of Setaria genome with its corresponding Phragmites genome, we can find that Phragmites has this stretch of genome twice in its genome. So this line is a genome, and the box in the line are genes. Each box is represents genes, and ribbons are connecting similar genes. So we can see that most of the genes, many of the genes are happening twice here, but not all of them are completely 100% duplicated. We can see that some of the genes, for example, this box is happening in once, but lost in here. And while others are just duplicated, but many of them are selectively kept in each copies. This is because perhaps when the Phragmites genome was first duplicated, there is like a plant scientist called it, say that the plant become a polyploid. And then at first, all of the genes must have been duplicated, but genome usually does not need any gene like in two copies. So many of them will be lost over the time, but only those that is beneficial for the survival of this species when it is kept duplicated, they will be kept. So there will be selective retention of duplicates, and that's why we are seeing only 35% of them duplicated here rather than the entire 100% is being duplicated. So now the next question is then what are these genes that are duplicated? Like what are the one that Phragmites genome is feeling that it is beneficial to keep it duplicated? So we can compare this Phragmites genome with all other grass genomes here, and then we can see that there are like duplicated genes this much in sky blue color. And when we compare this sky blue color with this orange color, then we can see what are the genes that is particularly kept duplicated in Phragmites compared to all others. And then what we found is that the duplicated group has a lot of genes that are regulating other genes, such as transcription factors. So these are not the one doing actual functions, but it is involved in signaling or regulating the function of other genes. So this is perhaps because it is easier to keep regulators in redundant copies for a genome, and probably these regulators provide some resources to when there is a certain environmental challenges, then quickly generate new gene responses to any changes or challenges in the environment. So this perhaps is the reason why regulators are particularly kept in this Phragmites after the duplication. And so far, we talked about the genome of the one invasive subspecies. To find out the difference between the invasive and the native subspecies, we need to sample multiple of each subspecies and compare. So that's what we did here. So across the Great Lake regions, we sampled 3 invasive subspecies and 3 native subspecies, often like growing side by side. Like you can see that the sampling sites are just 50 meters apart or 100 meters apart to each others. And we compare the gene expression profiles for these invasive and native subspecies. And one interesting finding we found is that the defense response function is always more enriched in genes showing higher basal level expression in the invasive subspecies. So invasive subspecies is expressing more defense related genes. This was a little bit surprising because we expected to see more differences in genes, for example, controlling the height or the growth rate or photosynthesis that is make the invasive subspecies bigger than the native ones. But the biggest difference we are seeing is in the defense related functions that is involved in the plant responses to other organisms. And this and also the transcriptomic responses to endophyte treatment, which I'm not going to show today, suggest that perhaps the interaction with endophytes or pathogens may play a role in the invasive behavior of this subspecies. So in conclusion, we found the whole genome duplication specific to this genome. And perhaps the endophyte and pathogen this interaction might have some roles in the invasive behavior of this Phragmites australis in the Great Lake region. And please check our paper for more details. You can check this QR code here. And we have been sequencing the genome of the native subspecies as well, and starting to find that the duplication, the invasive one, kept the duplicated genes related to defense and reproduction, and other like hormone-related ones as well. Yeah. With that, I would like to thank the funding and collaborators here, funding agencies and collaborators here, and thank you for the opportunity to present our work, and thank you for your attention.